steps, whether it's a skeletal structure or if it's a condensed structural formula or a Lewis structure. Always just follow the same steps when going to try to name it. Always the first thing to do is find the longest continuous carbon chain. So just start from all the corners and sort of try to figure out what's the longest possible chain. So remember that every end and every corner in a skeletal structure, that is a carbon, unless the end has a bromine, fluorine, chlorine, or iodine on it. So if you have the one, for example, like this one, just to show you that bromine there, that spot at the end is where the bromine is attached. So that wouldn't be a carbon at the end of that line. It would be a bromine at the end of the line. So that's like kind of the one thing that I see students like want to go, okay, well, that's a carbon right here. So that's not a carbon. That would actually be the bromine's bond. So the carbon above is the one that would be the one that you would have. So if you went through and counted them, what was the longest carbon chain? 8. So that makes this a octane. So this is an octane. So remember that. And you can always feel free to go in and just add a dot to represent each carbon, just to like help you remember. So you can see that that longest carbon chain in this one is the zigzag. It's not always. So don't forget that you got to check any other corners and such. Make sure that you check them all, because it's not necessarily just always in a perfect straight line. So there's the octane. So everything in there I have named. So this is a non-hydrogen group. So you would call this a bromo. This is a non-hydrogen group, and you would call this a ethyl. So this is a carbon and this is a carbon. So remember, a single carbon on a skeletal structure looks like a stick, kind of like the one above. It just sort of looks like it's a stick sticking up. But if it's got a bend in it, then that means there's 2 carbons. So this is an ethyl. So step 1, longest carbon chain, that becomes the last name in the molecule. Then circle and name any non-hydrogen groups. Then you've got to do what? Number left to right, right to left, or does it matter? You're going to start from the left, because if you go from the left, do you see that your bromine would be carbon 1, 2, 3? If I went from the other side, my ethyl group would be on carbon 4. So if you start from the right side and count in, it's 4 carbons before you get to the ethyl. So I would continue this 1, 4, 5, 6, 7, 8. And so that means my bromo is hanging off of carbon number 3. My ethyl is off of carbon number, 1, 2, 3, 4, 5. 5? Okay. It's fine. Just make sure that you double check. And that's why I like to number the entire chain, not like stop halfway along the way. So it's hanging off of carbon number 6 or 5. Now you have me saying it. So now I'm hanging off of carbon number 5. And then the last thing is to make sure that you put them alphabetical. So you're going to list bromo before ethyl. So this would be 3. Okay, pen. That's not a circle. 3 bromo, 5 ethyl, octane. And when you go to put these names in mastering, one of the things that mastering likes you to do is to actually not leave any spaces. So you would put a dash in between every number and every word. So it'd be 3 dash bromo, dash 5, dash ethyl. And ethyl octane would actually all be one word. So you would just write it without a space at all. Okay, so go back and practice those. There are a couple more at the very end of this chapter that we can do. Hopefully we get through all of chapter 4 today so that we have a little bit of time just to back up. So we kind of quit. We had started talking about isomers, but we left a couple of functional group examples off. So remember, we did these. So get your card. So when going about and looking at you're trying to identify functional groups. If you have a molecule like this, the first thing that I would look for is I would look for a sulfur. Look to see if there's any sulfur, because the sulfur is going to always have what name? You're going to call it it's always a thiol. So look at your small card, because remember that I will give you this. You don't have to write these groups on your note card. You'll have this, and if you find the thiol, anytime you see a sulfur, then it's going to be a thiol. Sometimes there's actually two sulfurs that may be bonded together. It's still, that's a disulfide, but it's still a thiol group. So that would be the easiest one that I would say you could identify first. Second one that I would look for is I would look at the rings. So look for the aromatics versus phenol. So how can I tell them apart? So first off, an aromatic or a phenol is always going to have a what? How many carbons in the ring? 6. So this is going to have it looks like a cyclohexane. So it's a 6-carbon ring. And the bonds are alternating single and double bonds. So it's going to be single, double, all the way around. How does an aromatic different from a phenol? It has that alcohol group. So it has an OH connected to the ring. So this looks like an aromatic, but it has an OH. So just look through the molecule looking for any 6-membered rings. Make sure that they're alternating single and double bonds. Then you know it's one of these. Then just check every carbon all the way around. If any of those carbons has an OH, then you know it's a phenol. If it doesn't, then you know it's an aromatic. So I think those ones are pretty easy to like pick out just because you're looking for that ring shape. Third one that I would look for is I would look at a nitrogen. So find nitrogens because they're going to be an amine or an amide. So if I find a nitrogen, I just put a new battery in here. If I find a nitrogen and any carbons around it have no oxygens, okay? So nitrogen could have 1 carbon connected, 2 carbons connected. It could possibly have 3 because remember nitrogen makes 3 covalent bonds. But if all of these carbons, any carbon connected to the nitrogen, if there's no double bond oxygen, then I know it's an amine. So look at your card. So do you see that? As long as the carbons connected to the nitrogen have no double bond oxygen, it's an amine. But if any of them, and it might be the one to the left, to the right, you really don't know, any of them though, has a double bond oxygen, then that makes it an amide. So you see the difference on the card. So when you find a nitrogen, don't just look at the nitrogen, look at any carbons connected to it, because that's going to tell you the difference between an amine or an amide. The oxygens are probably the most complicated ones. But you can kind of split this group up as well. You can think of oxygens at the end of a chain versus oxygens inside of the molecule. So if we have oxygens at the end of the chain, that means like there's going to be an oxygen like sticking off of the end. At the end of the chain, if it is a double bond oxygen with a hydrogen, then that's an aldehyde. If it's a carbon with a double bond oxygen and an alcohol group 2, then that's a carboxylic acid. So see those differences on your card? Those ones are going to be on the end of a chain. So end of the chain oxygens, look for whether there's 1 or if there's 2. It's going to be an aldehyde or a carboxylic acid. The other ones are inside of the chain. So that means that there's going to be like carbon, carbon, then a group with an oxygen, then carbon, carbon, carbons. So in the middle of a chain. If you see a double bond oxygen, so if you see a double bond oxygen, you kind of look to either side. So if either side is just a carbon, so just a carbon over here, nothing fancy, carbon on this side, nothing fancy, then that's a ketone. But if you see that double bond oxygen and there's an oxygen attached to that carbon, then that's the ester. but it's always going to be inside, not on the end. If it was on the end, it's a carboxylic acid. But if it's in the inside part, so if you have carbon, carbon, then an oxygen, carbon, double bond oxygen, then more carbons. So think of it as kind of being in the middle of the chain. That's going to be that ester group. And then the last one is if there is an oxygen in the middle of the chain. So you have carbon, carbon, oxygen, carbon, 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 just an oxygen acting kind of like part of the links in the chain, then that's the ether. So look at your chart, look at your card. So I do have this. This is posted in Moodle in your exam 2. I did move it into exam 2. So this card, I'm going to really encourage you to download this card, open it up on your computer when you do the dynamic study module for functional groups, because you really want to be comfortable using this card to help find stuff, because you're going to get this on the exam. So I want this to be a little more meaningful, but these are kind of like the steps that I would look for. I would say like oxygen's probably the hardest one in terms of identification. So remembering, like, when I look at these and see aldehydes and carboxylic acids and seeing those, that they're like on the end of the chain versus the ether, esters, and ketones that are in the middle of the chain. The last one, the only one I didn't talk about, is the OH. Okay, so if you see just an OH attached to a carbon, nothing else, that's an alcohol. But that could actually be anywhere. It could be at the end of the chain. It could be at the middle of the chain. The kicker is that you're going to see, if it's an alcohol, then there's an OH on that carbon, but no other oxygens attached to adjacent carbons, no other nitrogens on adjacent carbons, is basically just all by itself. So that is the alcohol. And so that one's kind of an exception. It could be on the end of the chain or it could be in the middle of the chain, but it's just characterized as just an OH group attached to a carbon with nothing else like on adjacent ones. It's not part of the aromatics or anything else. Okay, so looking at this one now. So use these steps. So number 1, do you see a sulfur? No. So no sulfur. Oh well. So that was the first chance. So then the second one is, how many aromatics do you see? 2. Okay, so you see 2 6-membered rings alternating single and double bonds. So you know that they're either going to be an aromatic or a phenol. So what is the top one? Phenol. Does everybody see that? Okay, so I see that 6-membered ring, but there's an OH attached to it. That makes this a phenol. What about the other one? Do you see an OH attached to the ring itself? No. If not, then that is a aromatic. Okay, those are the only 2. So there's no other rings in this molecule, so we've named those 2. So now find the nitrogens. Where do you see a nitrogen? Okay, so next to the aromatic, oh yeah, right here. Okay, so look at the carbon that's connected to that nitrogen. Does it have a double bond oxygen? No. If it doesn't, then it's an amine. So this is an amine. Okay, so definition, no double bond oxygen on an adjacent carbon means it's an amine. Find another nitrogen. Do you see another one? Down at the bottom. So you see this one here. So now you look at the carbons on either side. Does either of them have a double bond oxygen? Yes, so that means this is a amide. Okay, so it's this whole group makes the amide. So you don't name them separately, because sometimes students are like, well, that's a ketone and then that's a nitrogen. But they're actually, it's one group, those 2 together. So find the nitrogens. Just look at any carbons connected. If there's a double bond oxygen, it's an amide. If there isn't, then it's an amine. So now, any other nitrogens? I was like, did I miss any? No? Okay, so that one's done. So we look for a sulfur, that's a thiol. Look for the 6-membered rings, aromatics or phenols. Okay, so you're kind of like systematically taking all the things that are not carbon out of there. Then nitrogens. Now we're left with just the oxygens. So find oxygens. You could start with oxygens at the end of the chain, because oxygen at the end of the chain are typically aldehydes or carboxylic acids. So do you see one? Next to the what? Okay, so next to the aromatic we have one. So here. So see how that carbon has two oxygens. One's a double bond oxygen, one's an OH. So that is a carboxylic acid. So you could just put C acid. Since both the oxygen, double bond oxygen, and the OH are both connected to that one carbon, you would name it together. Not try to say that there's that double bond oxygen different from the OH. Since it's all attached to that one carbon, it gets named together. Any other oxygens? At the very bottom. So see this one. Also on the end of a chain, this is a aldehyde. Right? So you got to remember, aldehydes are at the end of the chain. And so look at the image for the aldehyde. Notice that at the end there's an H, right? So it's C double bond with an H. So that indicates it's not going to go on. There's no other carbons beyond. That's on the end of the chain. Find another oxygen. There's one in the middle. So this one? Okay. So where's that one? So look at the carbon that double bond oxygen is connected to. Does it have any other oxygens connected to it? Yes. So that means that this is a whole group. If it didn't have any oxygens, it'd be a ketone, right? If it was just a carbon with the double bond oxygen, you see what the ketone looks like? That's just a single double bond oxygen hanging off of a chain. But since this one has an oxygen attached to that carbon, that makes this a ester. Okay, so you see how the ester looks, how it's a little different. It's got that extra oxygen in there. Any others? Right above the ester. So here's this one. Okay, so you look at the carbon that's connected to that OH. Does it have anything else on it besides hydrogens? No. If it only has an OH, nothing else on the carbon it's connected to, then this is a alcohol. Any more? So notice up there, all just carbons. The one below the ester. Okay. So did I put the amide on this? That was the amide. So here. So I found an oxygen. Again, whenever you find an oxygen, look at like what carbons it's connected to. They're just carbons, yes? Okay, no double bond oxygens on any of them. They're just carbons. So this is the oxygen spanning the chain. So this is an ether. I don't see any other oxygens, do you? We've covered them all, circled them all. All the rest of them are just carbons. So this one's different because it's not just a single carbon-carbon bond like all the rest of them. Does anybody remember what a double bond's called? It's an alkene. Do you see that one up in the upper left corner? Having a double bond in the molecule. This parent name would be whatever the number size of the longest carbon chain, but it would actually end in ENE. So this molecule, just having that double bond in there, would actually make it an alkene. Okay, so those are sort of the ways that I would go about trying to kind of identify, because I think it's sort of the simplest ones, the easiest ones to find, and kind of working towards the more complex ones. When you're looking for oxygens, like look at all the ends of the chains. If it's on the end, it's an aldehyde or a carboxylic acid. I mean, it could be an alcohol, but typically it's these two. If it's in the middle of the chain, then you've got to figure out how those oxygens are arranged to figure out if they're a ketone, ester, or an ether. So I got another one at the very end. We'll do one more practice in this one. So then we started talking about isomers. So remember that isomers means that you have two or more molecules that have the same molecular formula. They have the same number of carbons, hydrogens, bromines, chlorines, whatever. They've got the same number of atoms, but they may look different. They may be connected different. They may be stuck in a certain position. So the first ones we talked about were structural versus conformational isomers. So remember that structural isomers are put together differently. So that was this first example. Butane, 4 carbons in a row, versus 2-methylpropane, which is 3 carbons in a row in a methyl group hanging off of the middle carbon. Those both have 4 carbons, 10 hydrogens, but they're connected different. And no matter how I flip butane around, I can't make butane into 2-methylpropane without pulling things apart. I'd have to break and rearrange bonds to have that happen. Those are structural isomers. So that is what these ones are. Those are structural isomers because they have a different bonding arrangement and different names. And that's really the big one. The one though over on the right, if you see butane drawn in the Lewis structure and it's nice and straight versus seeing butane drawn and it looks like that 4th carbon is sticking up, it's still butane. The longest carbon chain is still 4 carbons in a row. And then I had them kind of drawn a couple of different ways in that skeletal structure. You could just have your zigzags in any direction. But remember, if it's still the same number of carbons arranged in the same way, they'll have the same name. And then you know they're conformational isomers. So we kind of went through these two. First example, structural isomers, 5 carbons. And you can kind of look at these and see they're all put together differently. So you can see the first one's 5 in a row, the second one's 4 in a row, and 1 hanging off. So you can see there's no way they're going to have the same name just from kind of comparing the shapes of the molecules. Whereas the bottom one was maybe not so obvious. The bottom one, when you looked at it, you might have said, oh, I think those are structural isomers. But when you went about naming, they had the same name. They both ended up being 1-bromo, 3-chlorobutanes, even though they kind of look different because of the way that it's sort of like flipped up and bent. So do make sure if they have the same number of atoms, you know they're going to be an isomer type. So then going through the naming for ones that might not be so obvious will help you make sure that you identify them correctly. So you tell me with this one. Are these molecules structural isomers, conformational isomers, or are they not related? How do I know if they're not related? What do I have to do? Count them. First thing to do is just count the carbons. Okay? If they have the same number of carbons, then the odds are that they're going to be isomers. So first group, carbons, how many? This is C what? C8. And then H. I think I heard it. How many hydrogens did you get? 18. And if you have a non-ring structure, all single bonds, no non-hydrogen groups like this molecule, you will always have twice the number of hydrogens as carbons plus 2. Because remember, like if you think of a straight chain, you always have a hydrogen up and down, up and down, up and down, and then you have the ones on the end. So that's why like you can take 8 times 2 is 16, plus 2 tells you 18. And if you count them, that'll always be the case, even if it's branched. Because remember that carbon always has to have 4 covalent bonds. So no matter how the bonds are arranged, as long as they're all single bonds, the alkanes will always have that formula. Twice as many hydrogens plus 2 compared to numbers of carbon. Not if they're a ring, that doesn't count, but if under any straight chain or even branch chain arrangement, that's how they'll always look. What about the one at the bottom? How many total? So it's C8, so how many hydrogens? 18 as well. So if you see that they're the same number, then I know they have to be related. I know that they're isomers. So now it's the question of naming. So what's the longest carbon chain for the first one? 6. Does everybody see this? Okay, so remember that it doesn't have to be straight in a row. You're looking for the longest continuous carbon chain. So 6 makes this a hexane. Okay, this would be a methyl. This would be a methyl. Remember that the CH3 is hanging off. So now I've circled everything, so that's done. Numbering right to left, left to right, or does it matter? It doesn't matter, because if I come in from the left, you see that carbon 3 would hold the first methyl, but if I came from the bottom, do you see carbon 3 would hold the first methyl? So it really doesn't matter. 1, 2, 3, 4, 5, 6. So you get to pick. So that means that that first methyl hangs off of carbon 3, second methyl hangs off of carbon 4. So it'd be 3,4 dimethyl. 3, 4 dimethylhexane. So looking at the bottom one, longest carbon chain's a little more obvious. 6. So it's also a hexane. So this is looking like they're the same. This is a methyl. This is a methyl. And they're on carbons number what? 3 and 4. Do you see that? So, and again, in this one, it wouldn't matter which side I numbered from. Either side I come in, the first methyl group is hanging off of carbon 3. Carbon 4 is the second methyl group. So this is going to be the same. So these are conformational isomers. They have the same name. 3,4-dimethylhexane for both of them. It's just drawn differently. So maybe you looked at it and said, oh yeah, I see that. But maybe you looked at it and you're like, no, those aren't the same. Sometimes it's sort of iffy when they're bent like that, like when you end up not having this nice straight line. So sometimes it's a little harder to tell until you get into the naming part. So that top one we talked about last time, and I had you count the carbons, right? So it was C8, and the other one you started counting and you got 9. Once you see that there's not the same number of atoms, you can just stop right there. You don't have to go about naming them. Once you see that they don't have the same molecular formula, then you just say they're not related and you can just move on. So don't waste time going through any kind of naming. What about this bottom one? And I really don't know why there's a chlorine there. Just sort of like fell on the page. So first count total number of carbons. I got 11. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. You're right. C10. 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20. I got 21 hydrogens and I got a chlorine. What about over on the other side? How many hydrogens are carbons? 6, 7, 8, 9, 10. Oh, 10. And so that means I'm going to have 21 hydrogens and I got a chlorine. So now I know that they have to be isomers. Okay? Just count the number of atoms. I know they have isomers. What's the longest carbon chain in the first one? I got 7. Does everybody see 7? So start from the left, go across, and then down. If you go just straight across, you get 6. If you go from the right and down, you get 6. The only way to get 7 is this. Everybody see that? So 7 makes this a heptane. And so now I've got this, which is a methyl. And I got a CH2, CH3. That's a... What's a 2-carbon group hanging off? Ethyl. And then I've got this. So this is a chloro. So the next question, would you see when you start numbering how they come from different ones? Like, so coming across, if I start on the left, methyl would be a 2. But coming from the bottom, do you see that chloro would be a 2? So in this, there's always like, well, which way do you choose? So if there's ever a question where they're both like have hanging groups and they're not exactly the same, do you know that if I number them one way versus the other, I'd end up with different number combinations possible? So what they always say to do is start numbering from the one that's first alphabetically. So if I have chloro and I have methyl, then I would start numbering from the chloro side, just so that chloro would end up with the lower number. It doesn't happen very often, but if you see that, and you won't see it on an exam, but if you ever see that, that's why they end up choosing that, because notice it, ethyl would still 2 further, methyl is still 2 further, so either way, it's going to be like 2, 4, 6, or 2, 4, 6 in either direction. So just to give them continuity, they would say to start numbering from the group closest to the end with the first alphabetical letter. So this one would be 1, 2, 3, 4, 5, 6, and then 7. So that makes this 2 chloro, 4 ethyl, and then 6 methyl. So going over to crossed, longest carbon chain's a little easier to see. It is still a heptane. Do you see that? So that one's straight down, a little easier to see. This is still a chloro. This is a ethyl, and this is a methyl. Which way would I start numbering? From the top, because chloro is close, is got the C versus methyl. Do you see these are the same? These two molecules are the same. So this would again be 2-chloro-4-ethyl 6-methyl heptane. These are conformational isomers. Same name, just drawn differently. So then we kind of finished up here. So we finished talking about like, well, what happens if you have this ring shape? So if you have a ring shape, the ring like locks any non-hydrogen groups into a position. If there's only one, so like if I had this, if I had cyclopentane and I had just a methyl group hanging off, if I had just a CH3 hanging off, I wouldn't have to indicate whether it's on one side or the other, because all I'd have to do is flip the molecule and put it on whichever side I wanted. So you wouldn't need to identify the position of a non-hydrogen group if there's only one in a ring. But if there's two in a ring, their orientation to each other is really what I'm more interested in. So in this one, the one on the far left, if the groups end up on opposite sides, and so remember that's kind of like this one looks like it's sticking up, this one looks like it's sticking down. They look like they're on opposite sides of each other. The one that is sticking up or sticking towards you, they always use the wedge. And the one that's pointing down or would be like pointing behind you is always use those little dashes. And there is like no maximum or minimum number of dashes. It's really just like your whim. It doesn't really matter. And they don't really have to like go in one direction or another, just so long as you have them there. Because I know like some of the chem draw things get kind of weird. But just having some of the little dashes, that's what indicates behind. So that one they would call trans, and that would still be 1,2-dimethyl cyclopentane. 1,2 because the methyls are on carbon 1, carbon 2. So remember, in naming those cyclos, you always start naming from wherever your non-hydrogen group is, and then you just go so that the other non-hydrogen groups have the lowest possible numbers. The one over on the right, though, where they're both wedges or both dashes, indicates that they're both hanging off of the same side. So you can see in this one is here, and this one's here, so they're both pointing down or both pointing away from you, so they use those dashes. If you had them so they're both sticking up, then you'd use those wedges. So just remember, cis has the same symbols because those non-hydrogen groups are hanging off of the same side of the ring. Trans are going to be the two different ones. We will talk about this again in the next chapter. We are really not going to cover this in this chapter. Even though they introduce lipids in this chapter, I really like to cover all of lipids in one chapter, not splitting it up. And so we're just going to talk about our first nutrient group in chapter 5. That'll be lipids. But we're going to come back to this cis and trans because fatty acids, or the long chains of carbon that you find in fats and oils, they can exist with these double bonds. And double bonds can exist either as a cis, looking like this, where the double bond, I always say it kind of makes it look like the carbon creates this little V, or kind of a U kind of shape. That would be considered a cis double bond. Or it can exist in a trans arrangement. So trans, the carbons coming off of the chain go in opposite directions. So we'll talk a little bit more about those. Really, this is kind of introduced to talk about it in terms of lipids, but I'm just push that all off into the next chapter. So now the last thing. So the last thing in chapter 4 is the fourth kind of isomer. So we had conformational isomers and structural isomers. Then we have stereoisomers, the rings. And that's really the only ones you're going to have to worry with. It's just like identifying those rings and whether they're cis or trans. But the last one, these are called enantiomers. So they still have the isomer. They still have that omer ending like stereoisomers and conformational isomers. But these ones are called enantiomers. And this is because of special groups that carbon atoms can have. So remember, we've said this a lot of times, carbon always has 4 bonds. So if it's an alkane, then they're going to have 4 single bonds, bond up, down, left, and right. So if you draw those Lewis structures, you're always going to end up with 4. Well, they found that if carbon has different groups connected, then they have a special characteristic so that their mirror image pairs are not identical. And they give you this example. So this example is these two pairs of molecules. One of them they call limonene. And so there's limonene that is found in oranges. So have you ever had like a fresh orange and you peel it? You know the smell of that peel? That fresh kind of orange smell? That would be the left molecule of limonene. But there's one carbon in this molecule that has 4 different groups connected. And so if we make its mirror image pair, your olfactory cells respond very differently to the mirror image pair. That type of limonene is found in turpentine. So turpentine is actually extracted from pine sap. So if you smell turpentine, turpentine is used oftentimes to like strip varnish off of furniture. So like this is like fresh, nice orange smell, paint stripper. So they're very, very different. Even though those molecules, the atoms are connected the same, they have the same formula, the atoms are connected the same, but it is specifically because of the one carbon in this molecule that has 4 different groups attached to it, and it creates mirror images that your olfactory receptors feel are very, very different. They notice one as orange and the other one, someone was telling me like turpentine, like oftentimes they used to use it kind of you would like get pine needles, pine bark, and boil it. And you would get the sap, and that's what you could then purify. And so that really strong, piney kind of smell is they extracted it, and that's what turpentine's sort of from originally. But very different in terms of the aroma or the smell. And here's the why. So the why is this little carbon that's right here. That carbon that's at the bottom of that ring, and it's not an aromatic ring, it's just a cyclohexene. So that ring itself, that carbon in that spot, when you make the mirror image pair, they don't fit down on top of each other. They don't have that ability to be superimposed. So they say if the molecule has non-superimposable mirror images, that is what creates this last pair of isomers. So this one is a challenge, not my favorite example of the enantiomers because it's sort of a challenge to pick them out. But let's look at one that's a little bit simpler. So this one that's down here is a carbon with 4 single bonds, but the groups connected above is an alcohol group, so it's an OH connected above. To the left is a hydrogen. Now below it is a methyl group, so it's got a CH2, CH3. And to the right is just a single methyl. So when you look, and you're not just looking at what the atom is that's connected to this carbon, we're looking at all of the parts above, below, left, and right. So in doing this comparison, this carbon has 4 single bonds, and every single group above, below, left, and right is different. One is a methyl group, one is an ethyl group, one is an alcohol, one is a hydrogen. So you see that? Even though they're carbons to the right and below, they're still different numbers of carbons, and that's what makes those groups different. So if I make the mirror image, so the one that I drew is over on the left of the mirror. If I make the mirror image, then those two on opposite sides, really the hydrogen and the methyl group look like they swap places. Can you see that? So the green ball is what the methyl group is. So when I make the mirror image of this one, my CH3 ends up over here, the hydrogen here, the alcohol group is still up, and the ethyl group, is still sticking down. So still, when I pick these two up and try to lay them on top of each other, do you see that you can match up the alcohols, the top, the blue, match, and I can match the ethyl groups, which are the red. Those above and below will match, but look at the ones on the side. Do you see how the green and the gray, they do not pair up? Now I could pick this up and I could flip it around and get the green and the gray to match, but what would happen to the red and the blue? They would end up being offset, right? So no matter what, I can't make them sit and actually match together. So this creates a pair of molecules. They have the same number of atoms. They actually have the same bonding arrangement. But because of that one carbon, they create this non-superimposable mirror images. So a carbon that does that, they call chiral. And so you can say it chiral, that's fine, okay? So a chiral carbon is any carbon that has 4 single bonds, and all of the groups above, below, and left and right have to be different. And it's not just the atom connected to that carbon, it's everything. Everything above, below, left, and right have to be different in order for a carbon to be chiral. So the word chiral comes from this Greek term which means handedness. And so that's sort of the how it got its name. When you look at your hands, you agree your hands are mirror images of each other. So that means like the pinkies are close to each other. So it looks like if you were looking in a mirror, your right hand looks like your left hand. I can't take my hands and lay them in any position to make them exactly the same. If I lay them this way, then my thumbs and my pinkies are offset. If I flip and make my pinkies and thumbs match, then my palm and my wrist and the ends of my fingers are not matched. Even if I flip it over, that's not identical. Okay, then I'm looking at the back of my hand and my palm. So that's still not exactly the same. So no matter how I move my hands, I can't make them lay on top of each other and be superimposable. So that's really what happens with the chiral carbon, is they're different above, below, left, and right. So when you make the mirror image, they're not the same. So how do I tell? So you're going to get a molecule with a couple of carbons, and you're just going to examine each carbon as you go across. Okay, so like look at every single carbon in the molecule. You've got to first find all the carbons that have single bonds. So that means if you see any carbons with a double bond, any carbons with any kind of double bond connected to them, then you know they can't be chiral. There can't be any double bonds connected to the carbon or triple. You cannot make a chiral carbon if you have any more than 4 single bonds. That's like the first criteria. Check and see. So if you see any, so like if you get a molecule, you can just go X off any that are attached to a double bond. Whether they're a double bond to a carbon or a double bond to an oxygen, any double bonded, throw those out. You've got to have 4 single bonds. Then the second one is you're looking at the ones that are left, all the single bond carbons. They've got 4 single bonds. First thing to do, look at the number of hydrogens connected. So if I have a molecule like this, so if I have a molecule like this, does this have 4 different groups connected to it? No. I wish I could draw an H today. There, that looks a little better. Okay, so do you see that there's 3 hydrogens? Okay, so if you see something that's written in a condensed structural formula and it says CH3, could that possibly be chiral? No. If you see one that's written with a CH2, could that possibly be chiral? No. So most of the things we've been talking about and naming, they're not chiral. Okay, most of the molecules, it's got to have 4 different groups. So if there is more than 1 hydrogen directly connected to the carbon, then there's no way that it can be chiral. So this one's not, this one's not. So remember when we were drawing like butane. All those are hydrogens. Are any of the carbons in butane chiral? No, because they're all hydrogen, hydrogen, hydrogen. All these carbons have more than 1 hydrogen directly connected. So that's the first thing that I do is I look for double bonds. I X off any carbons that have double or triple bonds. Then I go back through and I count how many have more than 1 hydrogen directly connected. I X off any that have more than 1 hydrogen directly connected. That usually just leaves you a few. So really, you've only got to kind of examine a couple of them. Most of them you're not going to even have to look any further at. If you see that there's one that say only has 1 hydrogen, well, it has 1 hydrogen connected. I see all 4. Now I have to look at everything above, below, left, and right. And you're asking yourself, are any of the groups the same? So in this one, are any of the groups the same? Yeah, so can you see that that carbon's got a CH3 above and a CH3 to the left? So this can't be chiral because it's got two groups, even though they're not hydrogen, but it's got two groups that are the same, so they cannot be chiral. The only way it's going to be chiral is if they're different. And you remember in lab, if you were in lab, we did this just to make it as different looking as possible. I did this. I put a carbon, a fluorine, a bromine, a hydrogen, and a chlorine. So like you look at that and you're like, yeah, 4 bonds. Yep, definitely all 4 of those groups are different. But remember, you're not just looking at the atom. If the molecule's bigger than this, you got to look at everything to the left, everything to the right, everything above, everything below. Not just the atom that's right next to it. So let's do a couple. First one, what do you think? There's none. Do you see every single carbon has more than 1 hydrogen? So none. There's none in this entire molecule. That carbon is not chiral because it's got 3 hydrogens. This one has 2. This one has 2. This one has 3. So you would say there's none. There are no chiral carbons. I can't make an antiomers from this. So look at the one that's over on the right. Which ones would you throw out right away? The what? The 2 on the outsides. Do you see the one here? Do you see that this is a CH3? Can't be chiral. Do you see that this is a CH3? Can't be chiral. So I know that those ones are no's. So the only one that it leaves me with is the middle one. So I do see 4 bonds. There's something up, down, left, and right. Above is an OH. Below is an H. On the left is a CH3. On the right is a CH3. So is this chiral? No, because I have 2 CH3s. So you would say none. Let's look at the one that's vertical down. This is a carbohydrate. This is really the reason why we talk about chiral, because carbohydrates as well as proteins have lots of chiral characteristics to them. Look at the first one. So the first one's not drawn out like a Lewis structure, but CH2OH. So what do you know? Why? 2 hydrogens, CH2. So if you see 2 hydrogens in a condensed structural formula, this would be a no. Because remember, CH2 means that I've got 2 hydrogens connected and then an OH. So that's what that condensed structural formula is telling me. I have 2 hydrogens, so you can cross that one off. What about the next one? Double bond. Nope. Right? It has to have all 4 single bonds. So anything with the double bond, that gets tossed out. So now look at the third one. So here's this one. To the left, I've got an H. To the right, I've got an OH. Those are different. Above is this, and below is this. Are those the same? No. In fact, if you count, yes, there's 2 carbons, but notice there's 3 hydrogens above and 2 oxygens. There's 5 hydrogens below and 2 oxygens, so they do not have the same number of atoms above and below. So this is a chiral carbon. And the only way that you're going to know that is to make sure that you look left, right, up, and down. Okay? Or if it's a crossed, just make sure that you do everything to the left, everything to the right, and what's above and below. What about the next one? See, which ones can you toss out? Yep. Do you see that I know that can't be? Anything with the CH2 or a CH3 in the name, I know that it can't be. So that'll help you limit a lot. You're really looking for something that's got 4 different groups. So what about those middle ones? Yes or no? They both are. So when you see CH, what does that mean? Where is that hydrogen? Where would it be if we were going to draw a Lewis structure? It would be sticking up. Do you see that? It's attached to that carbon, and you would actually take this hydrogen and think of it like this, right? But when we write condensed structural formulas, they list all the number of hydrogens next to it. But do you see that if I was going to spread them out and look above, below, left, and right, that hydrogen would go up. This hydrogen would be up here as well. So now, for that first carbon, to the left is a CH3. above is an H. To the right is a 2-carbon molecule. Below is an alcohol. Are all those different? Yeah. So that one is, yes, that one's going to be different. What about this one? When you look above, below, left, and right, I thought I changed it. Change this color. It's just easier to see if you have a different color. So this one, hydrogen above, CH3 to the right, OH is below, and this whole thing over here is to the left. So all of those look different. So this one, I would say there's two chiral carbons. I didn't actually finish the sugar. 2 bottom ones, 2 bottom carbons. What do you think? This one. Is this one chiral? The one I just boxed in red. Left, right, below, and up. Are they all different? Do you see that? So above, I got 3 carbon group above, an alcohol group to the left, a hydrogen to the right, and a single carbon group below. So all of those above, below, left, and right look different. Yep, that's chiral. What about the bottom carbon? Why? 2 hydrogens. Do you see CH2? If you ever see an H2, automatically knock it off, right? If you see an H2 or H3, you know that there's no way. Or if you see 2 hydrogens directly connected to a carbon or 3, you know that they can't be chiral. So this one's a no. So that one I would say has 2 chiral carbons. That means I could actually, so that my mirror image, I could make a couple of mirror images. I could make the mirror image of the blue boxed carbon or the red one, because both of those would be able to make enantiomer pairs. So I could end up with 4 just from that. So what is the significance about this? So yes, we are going to talk about carbohydrates. We are going to talk about amino acids and proteins and the fact that they exist as these chiral molecules. But think about biological receptors. So when you take a medicine, typically that medicine is going to have an effect on some group of cells. If you're taking a decongestant, it is going to decrease the secretion of mucus from cells in the upper respiratory cavity, so that then, you know, it sort of dries you up. Well, it is actually going to block mucus secretion. So a decongestant has to interact with the surface of those molecules to be able to affect how much mucus they're secreting. The shape of the molecule is important. And the best example I can give you is like, so if we shake hands, you always put out your right hand. So you shake right hand to right hand, right? So turn to your partner if you want. You can shake right hand to right hand. Then what I want you to do is I want you to shake right hand to left hand. And how does that feel? Very awkward, right? So it is comfortable right to right, okay? Right hand to right hand. But if you have a chiral carbon, this is almost like you're trying to shake your right hand and their left hand. And it's like the backs of your hands come together. You're like, like it's very awkward. It's the exact same thing for biological receptors. Biological receptors recognize one type. They recognize like the right-handed version. The left-handed version just doesn't fit because it doesn't match up to those receptors exactly. And that's the reason why many medications, if they have a chiral carbon, only one version actually works. So the Food and Drug Administration actually now has a rule that any time you have a chiral carbon in a pharmaceutical, in any kind of drug that's going to be given to living things, if there's any enantiomers, any chiral carbons, you have to produce both and you have to test both forms to make sure that they're not harmful. So here's a couple of examples. So one is for a medication that they developed called DOPA that they developed for Parkinson's. So just a little bit about Parkinson's. So Parkinson's disease, it develops because of a lack of dopamine in the brain. So there is one little part of your brain that makes all the dopamine for the entire upper central nervous system. So dopamine has a couple of jobs. One of the biggest jobs that it has is it helps to coordinate fine motor control. So I think of people that like do really fine like needlework or paint like really tiny marks. Me, I have a hard time staying in the line. But for people that can do that, it's pretty amazing to me just how detailed some things can be. They have this ability, very strong fine motor control. You have to learn fine motor control just to be able to write. And people that have terrible handwriting, maybe that's like a challenge. Dopamine does that. So it helps with fine motor control. So when people develop Parkinson's, one of the symptoms they first notice is what? That they start a tremor. Okay, so you'll notice like a hand tremor. That is because of loss of that fine motor control. So you can't keep the muscle still and control how it moves. So instead, you're more jerky with your motions. Unfortunately, dopamine also plays a role in mood. So people that have Parkinson's oftentimes have depression, that they can have mood swings that occur because dopamine along with serotonin all factor into overall mood. Dopamine also has a role in memory. So oftentimes people with Parkinson's also develop some form of dementia. So seeing things, not being able to remember short-term versus long-term, staying in the times type of thing, all of those end up being these side effects, all because this is central nervous system affected. So not only does it affect its fine motor control, but it also ends up having these other roles. So it's a really big challenge for families to try and care for somebody that has Parkinson's. So some of them, there are meds that can add to it or that can actually cause like Parkinson's-like effects. And so some of it can end up having an effect on dopamine levels. And so you're trying to stop or block one problem, but it may end up having a side effect of actually triggering another type. So Parkinson's, though, as it progresses, if you lose dopamine completely, you lose motor function. So these people get less and less interactive, less and less mobile, and eventually they reach sort of a vegetative state. So they're not dead, they're still alive, but they're not interacting because of no dopamine whatsoever at all. So they said, well, we need to try and get dopamine into the brain. The problem is dopamine as a med will not cross the blood-brain barrier. So this is a barrier that's made up of cells. It's very selective, and it only lets some chemicals pass through. And it's a good thing because you don't want things randomly going in and affecting your brain because that controls everything. So that barrier would not allow dopamine to go in. So they found a chemical that is used by your brain to make dopamine. And that's where this drug is. So dopa is kind of, they call it a precursor. So your brain can take this drug and make dopamine from it. So initially, they were like, oh, this works great. So they gave people dopamine. The unfortunate thing here, this is your carbon. So remember, this would have 4. There's your carbon. I've got that aromatic phenol alcohol group on the left. I got a carboxylic acid group on the right, hydrogen up, and the nitrogen group below. Can you see that that is a chiral carbon? So the one form, so they called this L-dopa, and I always think of L, they named them L and D, which is like left hand and right hand, but it's from Greek, so it's levo and oh Lord, what is D? Dextro, I think. But the other one, I always think about them as left and right. It's not, it's levorotary and I'll think of it later. But then, so what they found though is L-dopa crossed the blood-brain barrier, got picked up by these cells in that little part of the brain, and they made dopamine. And these people suddenly woke up. They suddenly became mobile. They suddenly could respond to you. So they went from being almost in this persistent vegetative state into being completely functional. The problem though, they found D-dopa does nothing. So this one actually, this is biologically active, but this has no effect at all. And there was some frustration because, again, it wasn't really picked up that that was a chiral carbon. And so they were like, I don't understand. Some of our trials, nothing happens. Some of our trials, it works great. And they went back and looked at the chemical structure of the drug they were synthesizing, and they were making, if they made a mix, didn't have very good results. If they used just exclusively the L form, it worked great. And so that became, with the pharmaceutical companies, like kind of like a light bulb. We've got to make sure that we check all of the chemical structure of molecules. So albuterol is a bronchodilator. It has a chiral carbon. Same thing with ibuprofen. Same thing with naproxen. Same thing with Tylenol. They all have chiral carbons. And one form is biologically active and the other form does nothing. So when you go to do synthesis, you've got to make sure that you're producing the right form, just because it's a lot like this. Giving you the wrong form would have absolutely no effect. It would be like your right hand shaking your left hand. It would have no effect. It would not be recognized as normal and not bring about the biological change that you really want. Unfortunately, there have been instances, probably the most famous is a drug that was produced in the 1950s called thalidomide. So thalidomide, and you can actually see in this molecule, they show the skeletal structure as well as showing like the little balls and sticks. Thalidomide was developed to treat morning sickness. So if you're pregnant, usually morning sickness is when? In that first trimester. So it's typically in the first 3 months of pregnancies. It's very much related to like hormone changes that occur. It just causes excess acid secretion. But now there are some women that have morning sickness. This is really severe. So this can be where they start vomiting and they can't stop. Okay, so it is like an all day. They're not just sick in the morning. It's a morning, noon, and night kind of thing. And so they can lose weight. Well, so that if weight loss by mom is not good for baby. Okay, so there was this question of, well, what can we do to try and alleviate this? So this drug was developed, and they initially didn't know that it had a chiral carbon. So they synthesized the L form, the left-handed form, and they tested it, and it had great effects. It helped to reduce morning sickness. Women lost less weight. Women were healthier overall, so they would have better birth weights. Okay, because again, remember, this is 1950s. There's no ultrasounds, right? We didn't have any of the Doppler kind of stuff going on. You were basically like, yep, you're pregnant because you're getting big. That was like almost, like we can manipulate and feel baby, but it's very like call the midwife kind of thing. Okay, very, very like limited. Some of the things you watch, you're like, oh my God, I can't believe that's how they like had to do things back then. So it did help. So that went through. This happened in Europe. So this was really in Germany, was where this was developed. Once it got approval, they scaled up. So when you do your initial trials, you might test on 10 women. Well, first you test on animals, then you test on women, then you test on like an upscale. So they said, okay, this is safe and seems to really be helpful. So it was just given out. So anybody that went in the first trimester, doctor said, how are you doing? Well, I've been having morning sickness. I've had trouble keeping food down, blah, blah, blah. Well, here, you can take this new medicine called thalidomide, and this really helps to reduce morning sickness. But when they went to large scale production, they actually made both enantiomers. Because like when you do like small batch of things, it's very easy to control. But when you go from making like a pound of this to making 1,000 pounds, as when you increase the scale of production, a lot of times you have to change. You have to change like your recipe a little bit to make sure that it ends up working. And so, but there still wasn't this understanding of these consequences of chiral molecules, 1950s. So it was like 70 years ago. And so when they scaled up, they made this 50-50. Unfortunately, while most enantiomer pairs, one is biologically active, the other has no effect, the mirror image, the other enantiomer, is teratogenic. And teratogenic means has the ability to cause birth defects. So there was an estimated 10,000 babies that were born in the 1950s that ended up with severe limb deformities, because what's happening in that first trimester? So you go from being one fertilized egg, right? That starts day one. And in 3 months, you have a brain, you have a spinal cord, you have a ventral body cavity, you have arms, you have legs. And what thalidomide did was it stunted elongation of the buds that become your arms and legs. And so they ended up, you had all these babies, they called them like little flipper babies because a lot of times their hands were actually on the ends of their shoulders. So they didn't end up developing arms, but they would end up developing some kind of fingers. Same thing with legs. Only about half of them survived because unfortunately in the pelvic floor, like where your leg buds would start, there's a lot of organs that have to come together. So you have to have like the urethra and the anus. So all of those urogenital openings. So there were problems with pelvic floor quality for a lot of the babies. And so they didn't survive. So then you're like, how did 10,000 babies get born? That seems like an awful lot before somebody would be like, oh, this is bad. But think of when women took it. They took it in their first trimester. When did they see the birth defects? Not for another 6 to 8 months later. Okay, and so in that period of time, more women had been prescribed this because it did have very beneficial effects of cutting down on that morning sickness. So once doctors saw it, they would give it to the next women that came in and the next women. So this went on for close to a year. It was difficult to connect because it wasn't something they took like right at the end of pregnancy. It was something that was taken like early on. So it wasn't really recognized that it was actually because of this drug. So this drug stayed on the market for almost a year and a half.
Were they also still learning about the development of the baby?
So they kind of knew those types of things by that point, but it did take like some kind of sleuthing. Well, suddenly, this would happen occasionally anyways, but it was nowhere near the numbers that they suddenly started to see. So that is when they started to like question, well, why? What kind of medications? Was this, were you taking a supplement of some kind? Were you exposed to some kind of environmental hazard? Was there some other reason that this actually ended up? Because it was primarily in Eastern Europe, because the drug was originally like in this area close to the west part of Germany, and then was sort of like given out to France, given out to Spain. So all of these countries around had higher incidences. Because of this led to like some sweeping changes in terms of drug development. So now, if you develop a drug, one of the first things when you put forth your application for a drug that's going to be given to any kind of living creature, you have to know the chemical structure. If there's any chiral carbons, they have to be identified. And when you start your clinical trials, you have to produce every type of enantiomer possible and make sure that they're all tested. So thalidomide really ended up with this very, very bad, it's got a big dark cloud over it kind of thing. So they just took it off the market and no one used it. In the last 10 to 15 years, they have started using it because it really does have a beneficial effect of alleviating any kind of nausea. So who might have issues with nausea? Not just pregnant women. Think about like patients you take care of that are sick, that have nausea. Cancer treatment, right? So patients undergoing chemotherapy may have nausea and vomiting. So thalidomide would be beneficial in helping to alleviate those symptoms. But they only synthesize the left-handed form. They can control what's synthesized. That has to be tested to make sure that you're not synthesizing the right. And they don't give it to pregnant women at all. Okay, so those are kind of, and oftentimes patients going through chemotherapy, they are not pregnant women. Like that is like worst case scenario if that happens. But that's why they have started to use thalidomide as an anti-nausea med, but it is not given under any kind of questionable conditions because for anybody else, this had no negative effects. Yeah, well, they're just developing so fast. They're just like ridiculous. the amount of cellular growth and changes that happen with going from one egg to that 9-month span is just like craziness, All right. Okay. So I thought we'd do a few more. Well, it's almost time. We'll do, which one? Let's do that one. Okay. Draw or name. Okay. I have a couple of these, and again, like I did on the last chapter, I will go through and put the answers for any that are not. Remember that my marked up PowerPoints, they are posted in Moodle. The marked up ones are in the chapter 2 module, or chapter 2, in the exam 2 module, okay? The blank PowerPoints are in the PowerPoints for printing module. So those ones you could actually go back and practice. You could download the blank ones and go through and answer the questions. Then you can check the marked up ones to see if you can actually do them. That would be a great way to practice things like this, like naming ionic versus covalent, like drawing versus naming, identifying things like the isomers. So in this one, if I had cis-1,2-difluorocyclohexane, okay, how would I draw this? What would I put in first? 6 carbons in a chain and a ring. See the cyclo, okay? So remember, first thing is the parent chain. That tells you what to put down first. And so now I've got to have 1, 2, 3, 4, 5, 6 connected. That is cyclohexane. If you were going to do just the skeletal structure, remember that would be like a hexagon. Okay, cis-1,2-difluoro. So what does that mean? So I'm going to use what? If I want it to look cis. Okay, so I'm going to use either both dashes or both wedges. Where can you put the first fluorine? You pick, wherever you want, right? So it could be here. It could be there. It could be on any carbon, but I'm going to call that carbon 1 because remember with the cyclo, the hanging group needs to be carbon 1. And now how do I know where to put where I put the other fluorine? Just count. So I could put it on the bottom carbon, or I could even come back over here and put it on this one. As long as I use 2 wedges, then that would show me that they're both sticking out. They're both on the same side. So that would be carbon 1 and carbon 3. All the rest of those, remember that carbon has to have 4 bonds. So all these would be what's? Hydrogens, right? So in a ring, carbons always are going to have 2 bonds sticking off because they're always connected to 2 carbons in that ring. And if I was going to draw it just as a skeletal structure, then I could have the wedge, 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 or the dash, 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 because that's the cis. Just make sure that either one you use, it's the same, okay? If they were trans, I'd have 1 wedge and 1 dash. What about the bottom one? So what's the longest carbon chain? Yep, that's what I see too. Right, 1, 2, 3, 4, 5, 6, 7. So that makes this a heptane. Okay, this is a chloro. This is a chloro. And this is a one is a methyl, 2 is a ethyl. And I don't know why I like saying it that way. It helps you remember ethyl. It comes out of your mouth then. Ethyl, yeah. Otherwise, you're like, I don't know. All right, which side do you number from? Right or left, or does it matter? Right, because on the right is my chlorine. So this is 1 chloro, 2, 3 chloro, and then that's 1, 2, 3, 4, and then 5 ethyl. When I go to name it, which goes first? Chloros. So it'd be 1, 3 dichloro, and then 5 ethyl heptane. And remember that you would have a dashes. I don't care if you have the dashes. Like on the test, I don't have a problem if you don't have dashes. But in mastering, remember, try to put the dashes because they might like prompt you to do that. Okay, so I have that one. Here's another one. So I'll put the answers for these ones so that you can do them. So there's that one, and I'll also do that one. I don't really like the bends on that one, but all right. If you did not grab a note card, there is a note card up here in your Moodle page. I actually have like an extra review because some students wanted another review. So you can find those. So in here at the very top, I have like the regular exam review, and then I have like a checklist that says, can you do this? Can you do this? Can you do this? So maybe that gives you more things like, oh, I don't know if I can do that. Then you can go back and find them in the PowerPoints and practice them.